So I'm glad to introduce the next speaker for today. His name is Roger Conte. He has a biological background since he studied biotechnology for his bachelor at Químic de Sarrià Institute at the Ramon Llull University in Barcelona. Hopefully I pronounced Llull, no, exactly, I was sure about it. Afterward, he moved to Boston, where he took his master's degree at MIT, where he developed methodologies to study osteoarthritis knee cartilage function recovery from mechanical stimuli and also the medication administration. He's now part of this For All consortium as a candidate number 6, where he's studying the genetic underpinnings of the disease that also is intervertebral disc degeneration. And he has a special interest also in the application of machine learning to images and studying the limitation and the implementation of this technology to the study of the disease. So when the technicities are ready, I'm glad to introduce Roger.
So before introducing the topic, which by the way is disc degeneration, I'm going to give a simple vision of the medical research. We normally have a group of people that has an idea, a way how, or a product that is supposed to benefit a group of people, a community, which we're going to call the users. This is extremely easy to catch when we're talking about drug discovery, but it also applies to medical engineering and as well as software or human modeling as a twin human. Normally, the creators start to have in mind to deliver the product straight away to the users, but we know there's a lot of regulation that has to be obeyed. You have to convince the users that your product is better than the others. And that's extremely important with AI and modeling, where we have a lot of competitors nowadays and the publications are increasing. So what I'm going to talk about today is using my work. I'm going to explain or exemplify an alternative way to make an impact on clinical settings without deploying the product straight away. I'm not gonna, oh, it's moving as well. I'm not gonna enter too much on disc degeneration. We have heard a lot, but as we said before, it's mostly diagnosed using MRI. And from MRI, we extract different traits, and I just want to highlight that it's extremely complex and extremely heterogeneous. So once we have these phenotypes that's normally human and radiologist based, we can use twin studies to compare the inheritance between monozygotic twins and dizygotic twins and get an estimate of the heritability of the condition. In this degeneration, we have high heritability, which means at the end that we are supposed to have a lot of genes playing a role on the condition. How we do discover these genes, we normally use genome-wide association studies, where we get normally cases and controls or different phenotypes. We compare their genetic variants across the whole genome, and then we get genes or actually variants that are associated with the condition or the severity of the condition. And for this degeneration, we have really few GWASs, which lead to really few number of known genes that has an impact on the condition, and although having a high heritability. The reason because of that is normally because we are lacking analytical power. Since we are analyzing millions of variants across the genome, GWAS is extremely penalized by multiple test correction. So normally goes together with small sample sizes, and again, how well is defined the phenotype, how homogeneous is the phenotype for this association analysis. In order to tackle that, I started my PhD designing a multiple trait meta-analysis where we combined the genetics of the different traits of this degeneration. But still, it was not very successful. It was not very convincing. probably because we were using 2 cohorts, Twins UK you're going to see around, and Northern Finland birth cohort and FVC from now on you're going to see as well. They have a really small sample size, around the 1,500 1,500 samples. So this is a question, like, to increase the power, the analytical power, we have to increase the sample size and try to make the phenotypes more homogeneous. How are we going to do that? So the answer now is clear. Let's use the machine learning or artificial intelligence. Thank you. So the first thing that we did is run a systematic review with the meta-analysis of all the algorithms, all the papers that were published that were using machine learning to extract MRI features for this generation. And we compared the accuracy among other variables of the design of the study. There was, again, a lot of different algorithms, deep learning, random forest that were used. So we were comparing all of that, expecting to find application, a machine learning application that was better than the others that we could use afterwards. But from the meta-analysis, what we got is that it's not actually that important which algorithm you're using, but what's more important is that you need to have it validated in external cohorts and external source of data. And using these results, we decided to use, or we thought that the best application that's out there is SpiNet, because it's the one that has been most validated using different cohorts. So SpiNet, basically, it's just a deep learning application that extracts from the whole axial MRI. It isolates its disks, and it extracts different traits of this degeneration. So we did apply that on 3 cohorts, the ones I told you before, Twins UK and FVC. And we were lucky to be able to access to the MRIs on University of Michigan, which is, they have different, these cohorts are quite different on the sense of creation and structure. But they all have T2 MRIs. So we did apply this SpiNet to extract different phenotypes. Here I'm just showing, for exemplifying the distribution of each cohort of these phenotypes. And we can see that more or less they are comparable, and that's actually what we were looking for. As I said before, SpiNet is the one that has been validated the most. One of the validations was actually done in the Northern Finland birth cohort by one of our colleagues who is not here, unfortunately, and using other cohorts. Apart from that, I did do a small validation on Michigan because I've never been using Michigan. And as we can see here, there's a nice correlation between my grading and SpiNet grading. Again, that's something that we're looking for homogeneity. So I've been talking how to transform from the machine learning and the images to phenotypes. And I said that we're going to use genetics to go back to clinical settings. So I'm going to show you how this GWAS resulted in this data. As I said, genome-wide association analyses are very penalized by multiple test correction, and it's supposed to be used in big sample size, as UK Biobank Sina said. So that's the genome-wide significance. The green line, we call it a suggestive line. In this case, we are not still reaching genome-wide significance, but it's clear that we are getting a nice association that are really well differentiated from the noise from the rest of the variants that we assessed. And this is for herniation phenotype. Similar happens with Fermat score, which is similar to the KL score that Christina was saying, but for the disc instead of the knee. And plate defects that I think Paola said something about it. And with these analyses, we analyzed these peaks that were suggestive or significant. And we got a number of genes that are potentially associated with the condition. The good part is that we were able to replicate some genes that has been linked to the condition previously using other GWASs or using cell culture analysis. which is the case of SOX5, for example, similar with CHS33, which has been also associated with disc herniation and disc degeneration itself. But what I was saying, like, OK, we have genes now. What is next? So the first thing is that with this new approach of using machine learning, we were able to discover new genes. We're able to replicate genes that has been previously done. And at the same way that we were validating the biology of it, we were actually indirectly validating the machine learning application. Because the biology that comes out of the output of the machine learning, it's plausible. It gives an extra credibility on the machine learning. But as I said before, how do we transform these outputs that comes from machine learning to the clinical settings in a different way? So genes, apart from giving you explanation of the biology of the condition, can also be used to predict risk, as Christina was doing with proteins, but you can do the same with genes. In this case, it's called polygenic risk score, when at the end we compare the genes of each individual to the genes that has been associated in the GWASs. And we get a single output, which is, again, is a risk. How we're going to introduce that to the clinical settings? I'm going to make a change because I haven't finished my GWASs on disc degeneration. So we are using a GWAS for back pain, which is supposed to be correlated. I'm going to explain to you that there's different tools in clinical settings that are actually being used in hospitals. One of them is the StarBack tool, specifically in UK, it's used in UK. That is 9 questions that, according to the answers, you are classifying low, medium, or high risk of having back pain, chronicity of back pain, once you have gone to the clinic. So our intention is to integrate this already used tool with the polygenic risk score of back pain. Sorry, I could not do it with disc degeneration. And see if we are able to increase the predictability of this tool. This is unfinished, by the way, so I'm not going to be able to give you the final results. The PRS that I was talking about was created with UK Biobank, which is a big cohort with more than 500,000 people. And it was validated by myself in Twins UK itself. So we believe on this polygenic risk score. And I'm just going to show you the preliminary results of this association analysis. We can see that StarBack tool is extremely associated with disability. Sorry, I didn't say that, but we are comparing it with disability as days of pension scheme that has been taken off work because of disability. And we see that StarBack tool is associated, it's predicting disability, but as well, the PRS is also associated with this disability 2 years after of the questionnaire. I cannot show you more results because we are still making them, analyzing them, but we are seeing a small increase, well, small increase, we're seeing an increase of prediction of a machine learning application that combines both with other comorbidities. So I hope I was very probably fast and surface level, but I hope I show you there's not always a single way to get to have an impact on the users, in this case society. There's also another ways that creators can be users and can collaborate very well to give an impact on the users, the beneficiaries, in an indirect way. So that's mostly what I want to say. Thank you for all my supervisor, my group, all the cohorts, and all the other universities that were collaborating with me, and of course, participants of all the cohorts. Thank you very much.
Thank you, Roger, for this nice presentation. Very interesting. So I'm going to start to ask from the audience if there's some question. No?
Thank you for your nice talk. I actually just have wanted to clarify something. Maybe I missed it, huh? But can you go, I think, 2 or 3 slides back? Here, no, 1 4. this STBT and PRS, what does it actually mean?
Oh, sorry. STBT is the Starbuck tool. Is the tool that this one. This is Starbuck tool. Is the questionnaire of 9 questions that can predict chronicity of back pain.
Okay, and PRS?
Polygenic risk score.
Okay.
Sorry.
Yeah. No, it's fine. Maybe I just missed it because one was
I was fast.
explained.
I was fast.
Thank you.
Hello. Hello.
Yeah, thanks. Thanks a lot, Roger, for that. So, in the preliminary result that you have, it seems that life habits don't really have an impact. So, is that an impact on the Starbuck test result? Is
No.
Can you explain that?
Yeah,
a little
no,
bit?
don't get me wrong. As you can see here, the stars, there's some habits or comorbidities that has an impact. Like we know BMI has an impact on any kind of disability. Education and smoking, sometimes it happens. In this case, since it's disability of taking a day off work, we have occupational status as well as being significant associated. But we expect that as well, we know. But what's surprising is even having Starbuck tool in the equation, the PRS is also significant, meaning that they both combined can give a better prediction. And that's the end point. Does that make sense?
And then how do you see, then the genetic variance? Do you see it as a determinant factor for the fate, or do you see it as a risk factor that under certain circumstances it can favor?
Yeah, no, genetic variance, unless you're talking about a Mendelian disease, which is not the case.
No. Yeah.
It's always risk. It's never going to be determinant. Same as she was saying for the proteomics. It's always a risk, which has a lot of impact, has a lot of importance, because you can act according to the risk, which is also saves a lot of money. But no, it's never going to be determinant. And again, genes are something permanent that we can check when we are born, for example, which is happening to Scotland, with Generation Scotland. They genotype the babies because they want to make more studies and get a big cohort. But for example, until that's happening, which is not going to be soon, the idea behind is that this PRS now has probably, I don't remember, I think it was 1 million of variants, but you can always extract less variants from this PRS and make it maybe less accurate, but has less variants, and make a small microchip that you can just get a blood drop and get the genes of these specific variants, the variation of these variants, and you can easily predict the risk of having chronicity of back pain.
Sorry.
Thank you very much for the presentation. I'm not at all a genetic expert, but when you show the Manhattan plots, so you said there was none due to this penalization that you have statistically because of this. You don't have any signature that is really significant. So, but the PRS, you calculated it based on those that show like tends to.
First, PRS was not using these GWAS. It was using a back pain. Back pain GWAS that was done with UK Biobank. Okay. But again, anyway, PRS is never going to use only specific variants that are associated. It has been checked a long time ago, and it's not going to work because there's a lot of polygenic effects. And again, with very complex diseases, it's the summation of the small effects that makes a huge picture. What happens actually. There's many ways of doing it, but you get the most significant ones. The number, it depends on a validation process, that when you have another cohort, you test that and you check how many SNPs are the best prediction of the condition. Okay. And that's how you get the number of SNPs, which SNPs, the weight of the SNPs as well. Yeah. And then you get a single equation that relates to a final single score for each person.
There's a lot of research focused now put on that, and in OA, for example, for post-traumatic disease, for post-traumatic knee osteoarthritis, there are a lot of things moving on there. Thank you. Thank you for the explanation.
Time for one question. Yeah. So following on PRS, so PRS are proportional, like correlated to also the cohort size on the GWAS from which they are calculated. So basically, better GWAS is better PRS, better prediction. So it's always more or less.
More or less, because again, you have to be very careful when you try to move a study that has been done in a single cohort or a big cohort, whatever. But UK Biobank is UK based. If you go to Asia, that PRS is not going to be valid at all. Okay. Not at all. Because, I mean, normally with Northern European, which before was called white people, sorry, with Northern European people, ethnicities, it makes sense. It's going to work. Better or worse, depending on the niche population that you are, how well connected is going to be that city, but because, you know, how it works. Like, if it's well connected, it's going to be more ethnicity cross-matching. Yeah. But yeah, of course, it depends on the GWAS, yes, definitely, but not only on the sample size, also on the design of the cohort that you're using in the GWAS. That's why GWAS meta-analysis is the way of going.
Because
yeah, you bring together different populations, and that's the thing.
And from your experience, there is different behavior depending on the disease for which the PRS is calculated. Like, for instance, in low back pain, he has a certain predictive capabilities in this case, but in other conditions can have less or very high. Yeah,
there's two things that we have to understand first. Like, as I said before, the estimates of heritability, but not only the ones that we use with twins, that we calculate through twins, because this is always overestimating, but also how well the GWAS predict heritability. There's another way of estimating heritability. Again, if your condition is not very heritable, PRS is not going to do much. Mm-hmm. Mm-hmm. If you have high heritability, as here, it should do. The problem is that we don't have the power enough yet, but MRI is very expensive. A cohort that has MRI and genetics is even more rare. Hopefully in the future it's going to increase. Mm-hmm. And again, using this methodology, we don't need radiologists to grade the same way thousands of images. We can use it through MRI, machine learning, and that's the beauty of it.
Yeah, it's quicker. Okay. Another? Yes?
Yeah, Roger. It's a bit related, though indirectly, with Francesco's question. So when you're doing the GWAS, so are you so segmenting by age group? And what I have behind this question is, so as the test or the results then sensitive to possible epigenetic changes that are known to happen unavoidably with age?
Yeah, there's two things. We don't stratify by age normally. You can do it, definitely, but you don't have the sample sizing here. Yep. In other phenotypes, in other conditions, they do. I don't know, like anything that's taken from blood, you have big sample sizes you can do. And normally they do actually sex separate GWASs. Right. But what we do here is that we adjust the phenotype by age square normally. Not BMI, because it has some problematics, but definitely sex. So we do include so many confounders, we call it confounders, to the association analysis. Actually, a GWAS at the end, it's normally the regression model, and we can add so many covariates. So we do try to tackle that, adding as a covariates, but you're right. It cannot be linear sometimes. I'm working as well with fibromyalgia, which is pain. And we are trying to do GWASs, and it's not the same when you are comparing women from 0 to 40, and when they start to have the menopause, the linearity, it goes to nothing. So yes, age has a strong impact. We try to tackle adjusting for it, but until we know exactly how it affects, We can, certify, but we don't have the power right now.
So as a comment on that, so you've seen Paola's talk, so where Paola was showing that degenerate cells were basically not behaving as normal cells, and so according to the network modeling that Sophia then has developed, so some of you will work with her during the hands-ons. So we have incorporated in this proteomics, and we have seen that so these degenerate cells and these normal cells are not internally wired in the same way. So we call that intervertebral disc cells, or we can call that chondrocytes, but actually that's not anymore the same cell. So this different wiring, does that come from these genetic variants? So do we have had an epigenetic changes, meaning a permanent change in a gene variant that have arisen because of age, because of external conditions, or does it come out of other changes in the cells? We don't know. And actually in the project, so what we wanted to do was then so to make genotypic characterization of the cells that Paola has cultured and has presented. But it turned out to be challenging, time-consuming, costly. It couldn't fit within the project. So these are basically then the restrictions you have to cope with when you're doing then this kind of research. Then in order then to further link with what Roger has presented, SRUG, the work that Francesco will be presenting tomorrow, okay, so that you can see on Wednesday, sorry, so that then you can see how these different pieces so are basically or can be combined together, and so sometimes we've been able to combine, sometimes we haven't been able to combine because of the money restriction, time restriction, and the intention is there. And then based on these very complex multi-omics things that link nicely so with what Christina has presenting, what Paola is presenting, and also what Andrea has presented. So Andrea's a bit more at the forefront of what is being done commonly. So on so Wednesday morning, so we'll have then the talk from Natasha Pujou from Barcelona Supercomputing Center, who is actually then combining all these multi-omics with machine learning in order to find, explore functional links, and then explore this possible open question with pleiotropic effect of IL-4 age and epigenetic changes. So on those phenotypes, and then complete all this research. Yeah.
Thank you, Jerome, for this wrapping up. If there's no more question, I think we are on time, a little bit ahead of time to go to lunch.
Yeah. Very fast.
And to close the morning session. So thank Roger, thank to all the speakers, and